science test that is available on the Pearson website. The reason why we didn't have you each individually do it is because it takes a lot of downloads and updates on your computer, and we just wanted to avoid all that confusion, and I'm just going to take you through these 18 questions. Now these questions are just not all that the MCA science test is going to be over subject-wise. It just, we wanted to go through it to go over how the test is set up and how it looks, and just an idea of what kind of subject matter you'll be asked about. So each section, they'll tell you a little bit about what it's about, and there'll be a series of questions for every little section. So this one's about pea plant genetics. So I'll just read it out loud. A scientist prepared a slide of cells from a pea plant leaf, and here's the picture over here, and the question is, the scientist saw this plant cell when she looked through the microscope. Choose the part of the cell containing the genes that help determine most of a plant's phenotype. That's the way it looks, such as flower color, seed color, and height. So you just literally have to select what part contains the genes. The ribosomes do not, the chloroplast does not, the cytoplasm, that's the liquid portion, cell wall is the outside, the nucleus contains that, so you just pick it and press next. Okay, so here's the question. What is the expected result when mitosis is complete? The key word in this sentence is mitosis. We look over at the picture. This is just a stage of mitosis. This is actually metaphase when the chromosomes are lined up in the middle. So when one round of mitosis is complete, what do you have? If we remember mitosis, it produces cells that are genetically identical. So we can automatically eliminate B and D because it doesn't produce genetically different cells. So we're left with A and C. And then our choice is, do the cells have half the chromosomes of the parent, or do the cells have exactly the same number of chromosomes? And we should know that it produces cells that are exactly like the parent, which would mean that they would have the same number of chromosomes. So the answer is C.
Inherited. Seeds from the cross were planted, and the number of offspring having each flower color was determined. Her results are shown in the table.
So as you can see, some parts of the science MCA test has some, you can actually have it read to you, or some of it will be able to play, and you'll be able to listen about scenarios. So there's a scientist that planted pea plants, which we talked a lot about in genetics, and these are the parents of the cross, a purple flower pea plant crossed with another flower pea plant, and these are their offspring. This is what the children look like. Let's look at the question. In this cross, the scientists predicted that the offspring would be a 3-to-1 ratio, 75% purple and 25% white. That was just the 3-to-1 prediction that she guessed would happen, but she controlled all the variables that she could to get the result. Which is the best explanation of the source of error? So that just means, why didn't she get exactly 75%, 25% in her results? Why was it 71% to 29%? Was it her technique used to count the offspring? Probably not, because it's easy to count. Was it chance during fertilization of the ovules? Remember, ovules are the egg cells, and plants have those, and chance is the key word here. We'll come back to that one. The growing conditions for the offspring, well, hopefully she controlled for all of that, and they all had the same amount of light, the same amount of water. So that probably isn't a source of error. And parents using the cross, now that wouldn't be it at all, because she used the same parents. So the choice, the thing that caused the error, the reason why this isn't exactly 75-25 is just a chance. You know, Punnett squares predict probability, but that doesn't take away the factor of chance, so it would be B. Here you can see we have the same scenario, and we just have a different question. Which process best shows how the information in flower color genes leads to flower color? So this is just the order of things, the central dogma. DNA, remember DNA starts out in the nucleus and holds the master plan, the instructions. So automatically we could eliminate it to just A and C because it always starts with the DNA, which is located in the nucleus. Then the DNA has to be transcribed into something, and this also happens in the nucleus. And it's transcribed into RNA, not to protein, so that would be our answer. DNA is transcribed into RNA, and then it's translated into a protein, and that's what produces the color. So the answer is A there. Same scenario again, another question. A heterozygous parent and a homozygous recessive parent have offspring. Which question about the offspring is not answered by Mendel's laws? A few things going on. You need to know what heterozygous means. Heterozygous means one capital letter with a lowercase letter, and homozygous recessive means two little letters, so that would be like two little f's. And then we need to know Mendel's laws. Remember Mendel's law, one is the law of dominance, law of segregation, and law of independent assortment. So pretty much a lot of confusing things, but it's actually not too bad. Mendel's laws, all of his laws, they predict probabilities of many children. Like when we do a Punnett square, you predict the probability of 4 children with a simple Punnett square, not an individual. You cannot predict the phenotype, or what it will look like, or a genotype of any individual offspring. So the answer would be B. Mendel's laws do not figure out the phenotype or what any certain individual will look like. So that's the only one that makes sense. Okay, new scenario. A capital F represents the dominant gene, that's principal dominance of flower color, and the lowercase f represents the recessive gene. Okay, it's good to read that. Use the data in the table to determine the most likely genotypes of the parents. So this is the parents. We know their phenotype, they're purple, but what is their genotype or their combination of alleles? These are the different choices. Parent 1 could be this and this, but if the parents were both like this, they would not be able to produce a white flower, so that can't be it. They could be homozygous dominant and heterozygous, but that wouldn't be the case either because this parent can only give a capital letter and only produce children with purple flowers, so that wouldn't be it. You can drag and drop and just see how it looks. If both parents were homozygous recessive, both having little f's, they would only be able to produce children with white flowers, so that couldn't be it either. So the only option is for both parents to have a heterozygous genotype, because then they could produce flower children with purple flowers or white flowers. The capital letter F represents dominant, gene for flower color, and lowercase f represents recessive. So what is the probability that the first offspring of a cross between two parents with those genotypes, big F, and little f, will display the dominant trait? So you would set up a Punnett square and figure out how many of those children would have purple flower color. Well, it's actually not even that difficult. This parent right here with the 2 capital F's can only give a capital F. That means all the children would be purple, so it's 100%. Next little section, photosynthesis investigation. Okay, which statement is an appropriate hypothesis for this experiment? So a hypothesis is a statement. It's very clear, gives only one alternative, or it clearly states what your experiment's going to be testing. So let's read. 2 students prepare an experiment to investigate how the intensity of light, so how close or how strong this light is, affects the rate of photosynthesis in Elodia. That's a plant underwater, an underwater plant. They will conduct the experiment in a room without windows. So what would be an appropriate hypothesis? The distance from the light source to the Elodia should remain the same. That isn't a hypothesis. It's not a if-then kind of sort of thing. That's a statement. That doesn't make sense. As the light intensity increases, so as you increase the amount of light or the strength of the light, the rate of photosynthesis will increase. To me, that sounds like a hypothesis. Let's just read the other ones to be sure. The students will measure the rate of photosynthesis by counting bubbles. Yeah, but that doesn't tell you what they're testing. That's just how they're going to get the results. And D, experimenting in a room without windows helps control the amount of light reaching the elodia. Well, yeah, but that is not a hypothesis, so the answer would be B.
A sprig of elodia is placed upside down in a test tube filled with a solution. The test tube is placed in a large beaker full of water, which serves as a water bath. Photosynthesis will produce bubbles of gas. The bubbles will eventually come out of the stem and rise to the top of the test tube.
Okay, so you can listen to it or you can just read. The students will use a light source during their experiment. The light source will be moved closer to and farther away from the plant to change the intensity of light it receives. The rate of photosynthesis will be measured by counting how many bubbles come out of the stem in 1 minute. Now, with a question like this, it has a lot of reading in one question. A better idea, rather than reading the scenario first, would just be going right to the question. Which of these variables would be most important to keep constant during the experiment? So what's going to be most crucial to keep everything constant so they really know if the intensity of light is going to impact the rate of photosynthesis? So let's read it. The number of bubbles produced by the elodia. That's actually what they're measuring, as they tell you up here. Photosynthesis will produce bubbles of gas, so they cannot even keep that constant, so that wouldn't make sense. B is distance of light from the test tube. Well, that doesn't really make sense either because they will be changing that. That is the variable that they will be changing. Next one, C. Temperature of the solution in the test tube. That could vary, and that might impact results. So let's come back to that one. D, time of day the experiment is conducted. Remember when they said the experiment, they will be doing it in a room without any other lights? So it'll be no windows or anything. So time of day would not impact this experiment in any way. So the answer would be C. That would be most important to keep constant. So they would probably keep the temperature of the water in the experiment relatively constant through the experiment. Okay, now look here. The students want to know what the bubbles coming from the plant, what they are. So to know this, to know what the plant's actually producing, we need to remember the photosynthesis equation. On the left, we have the reactants, or what a plant needs to start to actually make its products, and the products are what the plant produces. And this just shows you the trial and how the experiment was, how the data was collected. So bubble production at varying distances. Distance of the light in centimeters from the plant, 15 centimeters away, 20, 25, 30. And bubbles produced per minute, average number, 60, 50, 40, 30. So looking at this data table, we know that more bubbles were produced per minute when it was closer to the light. Now, this actually has nothing to do with this question over here. It's interesting, but it actually has nothing. You could figure that out. They want to know what was in the bubbles. What is being produced to make these bubbles? So here's our choices. I put them in the right spot. So reactants, what does a plant need to start with? We know plants need light. That's the energy. We also know plants need water. And plants need to take carbon dioxide in through the leaves. This arrow means then, what do they produce? They release oxygen